Hi, my name is Nikhil Copeling, and today I will be sharing figure two from a recent study in Nature that is relating how brain tumors are undergoing therapy from 3D printed biologically active matrices that over a long period of time can release therapeutics that will stimulate the immune system in the body. So let's get right into it, explaining how well this new treatment helps mice survive after brain tumor surgery. So usually what happens with glioblastoma, which is the name of this brain cancer that the study was talking about, is that even after doctors remove the tumor, tiny cancer cells are left behind. Um, think about how, like, after you cut a cake, there can still be crumbs left over. That's a good analogy to kind of explain how even after resecting some of these tumors, resecting means to surgically remove some of these tumors, you're still left over with a few cells that could one day grow into a full-size tumor, and your problem is right back where you started. So scientists ask, what if we could leave something behind in the brain after the surgery that helps the immune system to clean up all of those leftover cells left in our brain? So in this experiment, the researchers compared different groups of mice after tumor surgery, and some mice received no treatment. Some received an empty implant, so this little biological matrix that doesn't necessarily have any therapeutic inside of it. And then some had this matrix that actually has this immune activating drug. So that's what we're seeing in A, and that's kind of a timeline of what we're looking at when it comes to uh, giving treatments to these mice, and then over time, if they're getting over their first interaction with cancer cells, and then if they are getting over it, they're being challenged a second time to see if their body has created an immune response now, almost a immune memory to where it remembers these cancer cells and knows how to fight back against them. So that's what A is showing here in figure two. B here in figure two is showing something called a survival curve. So what this shows is how many mice are still alive over time. You can think about it as kind of tracking how long each group stays healthy after they get their surgery, after their resection, which is when they remove the tumor. So in the untreated group, almost all the mice died pretty quickly. In the group that received the empty implant without the therapeutic, it was only slightly better. That's that purple line you see. And then the green line is the mice that had the implant with the therapeutic in it, and they had a much better survival outcome. So this is great because we're seeing that this therapeutic really is able to help these mice survive after they're being exposed back to these cancer cells. Now let's go on to the next part of this figure, which is showing not just that this therapy is working, but how it's working. So if we look at C here, C is looking at whether or not the tumors grew back after surgery in each group of mice. Um, what D is showing is that it's kind of showing what's happening inside of the brain after surgery. And instead of looking at the tumor itself, it's focusing on the immune cells within the brain. And so that's what D is tracking here. It's kind of looking at survival, but also looking at the body's defenses at the tumor site. Now, finally, over here in E, we're looking at if this treatment is really creating that long-term memory that we want from this therapy. And so the mice that originally received the drug-releasing matrix did not develop tumors again, even when researchers tried to have that that second reintroduction of those cancer cells that you see here, second challenge. And so those mice were able to survive through that and get rid of those tumors. Now let's look at the very last few parts of figure two, where we're looking at some more detailed parts of this study. So panel F is going to zoom in on a specific type of immune cell called a killer T cell. And so what this does is a killer T cell is what's actually killing our cancer in the body. And so we're looking at with the therapy, do the number of killer T cells in the body actually increase? And we are seeing that that is the case for patients for mice who've received this treatment. Um, Now in G, we're looking at how many immune cells are actually in the location that we want them to be in, because it's cool if we have immune cells within the body, but we want immune cells to be concentrated at the location where we had that cancer resection, where we removed that cancer, to make sure that there's as much tumor police there as we could possibly have. Now, H is going to look at whether or not the immune activation actually stays or not. And so it's kind of looking at our immune activation over a long period of time. It's a very hard graph to read, but it's showing that this matrix that we're putting in that's releasing the therapeutic doesn't just last for a short time, but for a long time, and can defeat those cancer cells that may be hiding. Um, Finally, in I of figure 2, and I over here on the right side, we're looking at memory T cells and those special memory T cells that we have, and how that's related to our cancer our cancer mice's survival. And we're seeing that that is working too. Overall, this entire paper is very important, this figure in particular, for showing that the the treatment with the biological matrix is not only helping mice survive, but it's showing the way that it's happening in a very straightforward manner. And thank you so much for listening to my presentation. Where's the recording? Oh my God. How do I stop?